Alrighty. Plasma membranes of bacteria, prokaryotes, and eukaryotes are very similar. Phospholipid bilayer with proteins interspersed. Some organisms, like animal cells and mycoplasma, have sterols in them, like cholesterol, to make it tougher. Other organisms might not have the cholesterols in them, like plants usually don't. Plant cells usually don't have cholesterol in them. But overall, eukaryotes and prokaryotes, same kind of plasma membrane. Now, archaeobacteria, we didn't really study archaeobacteria structures that much because I don't care about archaeobacteria because they're probably not going to cause disease. But they don't have the same type of a plasma membrane, so they're different. So it's just bacterial cells and eukaryotic cells have that same plasma membrane. All right. Okay. Don't forget your microscopy.
Oh no.
Don't forget your parasites. They're going to come up. You might get different questions about them.
Like what?
Like what kind of microscopy would you use to look at a live specimen in a drop of water because you want to see it swimming with its flagella? Probably phase contrast or something like that. What if you want to look at bacteria in a stained smear? Bright field, and what magnification would you use?
Oil immersion.
Right, oil immersion, which is how much magnification? Total max.
1,000.
1,000, good. So you'll get questions like that. Different kind of organisms. Don't forget your different kind of organisms, and don't forget your different kinds of microscopy. Okie dokie. Don't forget the steps of your Gram stain. Gram stain is going to come out purple or pale red. How did that happen? First stain is called the primary stain. What is it? Nope. Crystal violet. Purple. Then, why did you add the grams iodine? Mordant for what? It sets the stain. It makes that purple sticky and stick in the peptidoglycan better. Then what's your third step, the determinative step? Decolorizer. What'd you decolorize with? Ethanol, alcohol. The alcohol is either going to decolorize the cells if they have thin layers of peptidoglycan, and they'll be clear. It cannot decolorize the thick layer peptidoglycan. They're going to stay purple. And then you counterstain. Why?
To see it.
Contrast, right? Most of the time when you're staining something, you're trying to provide contrast. You want to make sure you see your cells against the clear slide with the bright lights on. So the counterstain is red. The gram-negative cells will pick up the counterstain. The gram-positive cells are not affected. They stay purple. Okay, so sometimes you get a little thinking questions. What happens if I stopped at this stage and put it on my microscope? What would I see? Or what would happen if I stopped at that stage? Or what would happen if I only had this kind of bacteria or that kind of bacteria? What would happen if I had a mixed culture? So as long as you know the steps of your Gram stain and which one's supposed to come out red, which one's supposed to come out purple, you should be able to mentally navigate the question. Alrighty, don't forget your trees of life. Remember your trees of life? You have the old tree of life, had 5 kingdoms. Animalia, Planta, Fungi, Protista, and Monera is all prokaryotes. You have the new tree of life, Carl Woese. Carl Woese said, hold the phone, we did that wrong, let's change it. We need more than 1 kingdom for bacteria. In fact, we need 2 domains for bacteria. And we'll put all the eukaryotes in the 3rd domain because there's really not as many of them as there are bacteria. Okay? Let's talk about classification of bacteria now. The way we classify bacteria with our Burgess list, Burgess manual is a manual of classifying and identifying bacteria. How did we classify and identify our bacteria upstairs? We looked at their shape and arrangement. We looked at the Gram stain result. Does it have a thick wall or a thin wall? Does it have endospores perhaps, or is it spiral shaped, right? And then this week we're going to look at what certain nutrients it can grow on. Is it a facultative anaerobe? Is it a strict aerobe? So those are all things we can see in the bacteria and can do visual lab tests. That's phenotypic classification. Carl Woese could sequence the genome of bacteria. And he actually didn't use bacterial DNA, he actually uses bacterial RNA, if you read your book at all. And if he uses DNA or RNA, he's using genetic material. So what type of classification is he doing?
Genetic.
Genotypic. So phenotypic is how you look. Genotypic is what does your DNA and RNA look like? What are your actual genes? So he actually used RNA from ribosomes in order to sequence bacteria's genomes. So, and Burgie's manual has been updated to include that information. So Burgie wrote his Burgie's manual like, you know, in the 1900s, and he's long since dead, and it's been revised. But the old way still exists, and then they added on new types of classification. So ours is phenotypic. Ours is based on phenotype. Genotypic classification of bacteria is how your textbook classifies the bacteria, and also Biomed Central, which is in your handouts package. They're very similar, have slightly different names. The genotypic, they use ribosomal RNA. Bacteria actually have RNA in their ribosomes, just like we do, and they could look at that. They found that to be more reliable than using their DNA. And the phenotypic, so let's put this as a Bauman is your textbook, and in your handouts, that's called Biomed Central. Actually, I like Biomed Central because it kind of is a blend of genotypic with the phenotypic, and then they put it in these nice tree graphics for you. Phenotypic, this is Burgie's, our way, I'll call it, our list. This is Burgie's old list. Let's say Burgie's original. How about that? Original list. All right, so phenotypic is appearance or morphology, more visual. There's something else I was thinking of, and it just popped out of my head. Oh well, it'll come back. Okay. Oh yeah. So you know what morphology is? Morphology, study of, you know what morphing is? You take like a human face and you morph it into a lion face. So phenotypic appearance is morphology. Whereas this is based on a genotype. Genes. Or whatever. On nucleic acids, RNA is not technically a gene, but it's a copy of the gene. All right. Okay, let's say you're gonna do a gram stain. Or maybe you're going to do a direct stain. What do you do first? You get your slide, you label the top of the slide, you sterilize your loop, you grab a little tap of bacteria and you mix it into a drop of water on your slide, and then you heat fix it. What's it called? What did I just make?
Smear.
A smear. I smeared it on my slide. I just made a smear. Good, that's a question. What do you call it when you get that little thin film of microbes on your slide? You call it a smear. In this case, it's a bacterial smear. Sometimes you'll get a pap smear. And a pap smear actually means they take a smear where they scrape off some of the cells of your cervix. Just like we scrape the inside of our cheek and we get some cells, well, they can go in there and scrape your cervix and get a few cells from that. And smear it on a slide. That's a pap smear. The reason it's called a pap smear is because the doctor who invented the technique's name is Papillon law. Whatever. It starts with pap. He has a funky name that starts with pap, so they call it pap smear. They named it after him. instead of calling it a cervical cell smear, or cheek cell smear, cervical cell smear. And then they look for abnormalities in the cells. Do the cells look like they're normal healthy cells, or they look somehow, and then they give them classifications, class 1, class 2, class 3, class 4 cells. Okay. A gram stain is a differential stain. What differences are we looking for? Differences in the what? The what?
Parents?
No. What about the bacteria makes the gram stain come out positive or negative? The cell wall. Good.
Yeah.
Acid fast stain. What about the bacteria makes it come out pink or green? The waxes. Whether it has the waxes in the wall or not. Right. And those are both differential stains. They're both differential stains. Okay. You did get the tannic acid question on your lab quiz, right?
Tannic acid?
In the staining lab quiz, you didn't get the tannic acid question?
Nope.
Okay, it's in the test.
The TB.
The tannic acid, what do we use tannic acid for, remember? The mordant for the flagella stain.
Oh, that one, yeah.
If that was in your lab quiz, good practice, it's on the test also. So you might get that question on your test. So that's why I say lab and lecture, it's all kind of mixed together. So you might get a lab quiz question on a lecture test, you might get a lecture topic on your lab quiz. That's related to that lab. Okie dokie. What was the mordant that we used in the gram stain procedure? Iodine, Graham's iodine. What's the mordant in the acid fast stain? What serves as the mordant in the gram and the acid fast stain? Remember, we didn't have to add one because it's already in the cell walls. It's the waxes. The mycolic acid waxes serve as the